Mutants are growing in a chemistry lab at the University of Regina. Fungal mutants, that is. Professor Tanya Dahms and her team of researchers use these specimens to study cell polarization. Undergraduate student Miriam Abbas looks at the differences between wild-type fungi called Aspergillus nigellans, common bread mold, and a mutant strain. The goal of this project, in collaboration with the University of Saskatchewan, is to understand how cell wall components contribute to fungal invasion.
Currently, I'm investigating a mutant strain of Aspergillus, and this mutant strain has a cell wall component that has been knocked out. The goal of our research now is that we want to find a treatment for Aspergillus fumigatus. So by weakening the cell wall, we hope that we can actually cause enough damage to the fungus that it won't be able to grow within humans.
In this lab, cell-to-cell communication has nothing to do with texting and everything to do with changes in the cell membrane. Professor Dahms explains.
The group looks at interfaces. We do research on lipid bilayers and cell walls. Internal versus external communication happens through receptors that act as antennae on the outside of the cell. So a small molecule will come along, bind to a receptor, and the message will be transduced inside the cell. One of the questions that my research group asks is how the physical characteristics of a cell change as a function of a drug or a certain stimulus, and that could be something such as an environmental toxin.
Undergraduate student Adam Clay is researching this topic for an honors paper.
I'm looking at the human mu-opioid receptor, which is the protein responsible for binding heroin and morphine and all of those sort of types of drugs. We're looking at why patients in methadone maintenance treatment, so opioid addicted patients, have such high variability in their optimal dose, and we think that it's due to their location within the plasma membrane.
Whether studying fungi or drugs, these students say biochemistry research is a great opportunity to look into the unanswered questions of science.